Neurons have to have a way to detect signals, integrate information, and then pass that information along to the next cell in a circuit. A key player at the cellular level, and how they actually accomplish this, is the movement of ions across the cell membranes. So in this video, what I want to do is I want to give you some basics about ion movement. What are the ions? How do they move across membranes? And how do they contribute to some of the basic phenomena that we're going to see in nerve cells? Well, let me begin by reminding you that neurons have different functional domains. There are dendrites, cell bodies, axons, and terminals. And for simplicity, I'm going to begin by just drawing a cell body, and then we'll add in the dendrites and the axons later. Well, what I want you to do is consider an experiment in which we imagine that we could take a tiny little voltmeter with two little leads and then measure the voltage between those two leads. Well, if I've got a cell growing in a solution in a dish, and I take those two electrodes and I place them both into the saline outside the cell, and then I measure the difference between them, I'd get a voltage of 0. There won't be a difference between them. But if I take one of those electrodes and then insert it into the cytoplasm of the cell, what we see is that there's a rapid drop showing us that the inside of the cell is negative. And a typical value that you would see would be -60 or -70 millivolts, more negative on the inside than the outside. And this would be true for neurons. It's also true for other cells. Pretty much every cell in your body is inside negative. Now, the vast majority of cells in your body spend their entire lives at this resting potential, and they don't change from it. What makes neurons and other excitable cells different is that they can sometimes change the voltage inside the cell. We'll come back to that in just a little bit. But first, you might be wondering, well, why are cells inside negative? And there are several explanations, or several contributors. One explanation is that the cytoplasm contains a large number of proteins. Proteins tend to be negatively charged, so that contributes to it. Another factor is that there tend to be more ions in the cytoplasm, and anions like sulfate and phosphate are negatively charged. But the biggest contributor to the inside negative potential is that there are unequal concentrations of ions on the inside and the outside of the cells. And there's also an unequal permeability of the membrane to those ions. Well, what do I mean? Well, to get at this, let's blow it up and look at a cell. So here's a little bit of a cell. We can see the cytoplasm and the extracellular fluid. And I hope that you recognize that this lipid bilayer, would be part of the membrane that's serving as a barrier separating the cytoplasm from the extracellular fluid. Now, you probably remember from a previous bio class that lipid bilayers are not permeable to ions. And if an ion wants to move across a membrane, it's going to need help, and the help comes in the form of integral membrane proteins. These integral membrane proteins take several different forms. For example, there is a prominent protein in nerve cells called the sodium-potassium ATPase pump. And this pump takes ATP, breaks it down, and then uses that energy to move 3 sodium ions out of the cell and bring 2 potassium ions into the cell for every ATP it breaks down. We're running this pump all the time, and what you can imagine then is if we were to go and look at the concentrations, we would see that outside the cell there'd be more sodium. Inside the cell, there'd be relatively little sodium. Inside the cell, there'd be lots of potassium, but outside the cell, not very much potassium. And in addition, because we're moving 3 sodiums out and only 2 potassiums in, this can help contribute to the resting potential. There are other kinds of transporters in the membrane that move chloride and establish a gradient for chloride. Typically, it's high on the outside, low on the inside. And for calcium, there's very little free calcium in the cytoplasm, and there isn't that much, but there's quite a bit more, relative more, calcium in the extracellular space. Okay, so now we have this situation, and what we want to know is, well, what would happen then if we allowed sodium to move, or potassium to move across the membrane? And this could happen if we introduced a protein that allowed their movement. To understand what will happen, we have to think about the electrochemical gradient, and that's exactly what it sounds like. It's the gradient for sodium that depends both on the chemical or the concentration as well as on its charge. So in the case of sodium, we have a chemical gradient that would favor sodium moving to the cell. There's 145 millimolar sodium outside, only 12 on the inside. It's going to move down its concentration gradient. The electrical gradient also favors movement into the cell because sodium is positively charged and the inside of the cell is negative. And what that means is that if we were to insert a protein or activate a protein that allowed sodium to move, it would move into the cell. The situation for potassium's a little bit less clear, and I'll walk you through why that is. So there's a chemical gradient for potassium to move out of the cell. There's more potassium inside than there is outside. But because potassium is positively charged, the electrical gradient would favor it staying inside the cell. Well, when you add the combined electrochemical gradient, what you find is that the movement of potassium out of the cell is favored. So it would actually move out of the cell down its electrochemical gradient. And this illustrates why it's important to consider the total factors that are acting on an ion. Now, if we go and we activate a protein and we allow these ions to move, the next question might be, well, how long are they going to move? And the answer depends on its equilibrium potential. So what is that? Well, if you think about what happens if you open up a channel, two things might happen. One is that the concentration could change, and the other is that the voltage could change. And in fact, what we see is that the movement of a small number of ions can cause a big change in cell voltage. So when we think about how to describe the equilibrium for ions, we talk about it in terms of a voltage. Because if we open up a channel and we allow just a handful of sodium ions to move through, there can be a big change in the cell voltage, and there won't be much of a change in the concentration. And the sodium will move into the cell until it reaches the point where it's at equilibrium, and it's gotten so positive inside the cell that sodium no longer wants to move in despite the chemical gradient. It turns out that for sodium, the equilibrium potential is about positive 55 millivolts. There's also an equilibrium potential for potassium, and for potassium, where potassium wants to move out, the equilibrium potential ends up being about minus 90 millivolts. Okay, let's go back to this figure. Well, I've shown you this before, but now what I've done is I've added the V equilibrium for potassium and for sodium onto this chart. And what I hope you can see is that the resting potential lying between minus 60 and minus 70 millivolts, well, that's closer to the equilibrium potential for potassium at minus 90 than it is to the equilibrium potential for sodium at plus 55. And what that tells us is that at rest, the cell membrane is more permeable to potassium than it is to sodium, because the cell is basically closer to where potassium wants it to be than where sodium wants to be. All right. Well, remember that I said we'd add back the dendrites and the axons? Let's do that here. Now, neurons can change the voltage inside the cell, and they'll do this in response to natural signals. But for this video, let's just consider an experiment. And the experiment that I want you to think about is, suppose that we had another electrode, and we also insert that into the cell, and we can use this to inject current. Well, if I inject a small amount of current, what will happen? Well, what will happen is if I add positive charges to the cytoplasm, the cell is going to get more positive. We call this a depolarization. If the cell gets more positive, it's depolarized. Now, in the case of a small amount of current that's being injected, we'll get a small depolarization, and it won't last for very long because the ions will diffuse and the pumps will kick in and they'll be removed. In fact, nothing really is going to happen unless we reach a certain voltage called the threshold voltage. And if we inject enough current to reach that threshold, so let's say I inject a lot more positive charge, well, under those circumstances, I'll get a bigger depolarization. And then what happens is instead of, just dissipating, we see this all or nothing big swing in voltage that I hope you might recognize as an action potential, the event that's initiated at the axon hillock and that can propagate down. What might cause the action potential? Well, we can make a guess now that we know about equilibrium potentials. What we see in the action potential is it goes from the resting potential and suddenly approaches the V equilibrium for sodium. And you could imagine then if we suddenly made the membrane more permeable to sodium, we'd shoot up towards that. And then if we suddenly made the membrane more permeable to potassium, then potassium would rush out of the cell to try and make it be minus 90. And in fact, that is what's going to happen in an action potential, and we'll see that in another video.